Last but not the least, we will have Dr. Oath to come speak with us. Dr. Oath has been an assistant professor in psychiatry department at the University of Pennsylvania for the past 3 years. He is a member of the Penn Medicine Translational Neuroscience Center, Neuromodulation Working Group, the Center for Neuromodulation in Depression and Stress, and the Neuroscience Graduate Group at Penn. He leads cutting-edge NIH and foundation-funded research that combines non-invasive brain stimulation with functional MRI brain imaging to discover new personalized approaches to the treatment of anxiety and depression. Currently, his laboratory is running a clinical trial using fMRI to guide TMS treatment of post-traumatic stress. So, welcome, Dr. Oath.
Thank you, Cedric, and thank you, neuroscience students, for inviting me. I'm very flattered to be on a panel with such accomplished scientists who are also my mentors here in my department at Penn. And so the theme of my talk is why hasn't brain imaging done more to kind of help in treatment of psychiatric disorders, but what I focus mostly on is brain stimulation and anxiety and especially depression since I've been at Penn, so I'm going to talk to you guys about that. I have disclosures. I hope to increase the number of disclosures the next time you guys see me. So this is just giving research talks at various places. Very once in a while you get paid to do that. So let's see. So beautiful brain images, right, of many, many thousands of volunteers and NIH-funded studies that say, hey, we think we have a really important diagram here of connections in the brain and how they underlie really important high-level functions and cognition and mental illness, really important work, but translating that directly into something that people use in a clinical situation, there's a gulf there. So when you go in to see your primary care, if you go in to see even your psychiatrist, and you say, hey, I've got this emotional problem, they're going to say, okay, tell me more about that, and maybe we'll do a little bit of cognitive testing or something like that, but chances are they're not going to order an MRI because it's not going to inform your treatment. Meanwhile, many of us are working really hard to look just in the brain imaging world to say, can we tell earlier on who's going to develop the disorder or what kind of treatment is going to be better for them? And so very important work, but at the same time, making that bridge to saying now we really have such strong evidence from the brain imaging data that now we're going to determine our treatment based on that. There's a sizable gulf in making clinical decisions. And I think part of the reason why is that psychiatric disorders are really complex and it's hard to tag one marker and really follow it and say we know what this part of the brain really causally does to other parts of the brain, and that's the place where you should monitor your intervention or where you should really pay close attention. So that's a problem with straight brain imaging studies. And in parallel, at the same time, we're doing brain-based treatments in psychiatry. We're absolutely doing that. So for 10 years, people have been doing this, approved by the FDA. You go into especially university clinic, private clinics, a lot of insurance carriers will cover this, and they say, let's use transcranial magnetic stimulation, non-invasive brain stimulation to treat treatment-resistant depression. So you tried some medications, they don't work for you, you're kind of desperate for something hopeful, and this treatment option is available to you. Unfortunately, the neuroscience world and this kind of treatment, which is a brain-based treatment, so you feel like, this is state-of-the-art, it's cutting-edge stuff, there's actually a big gulf between those two worlds that I hope with colleagues to bridge and make better use of this treatment. So you might ask, okay, so you have this stimulator and now you're going to decide where you're going to stimulate in an individual patient's brain. So probably you're going to, in that case at least, right, you're going to take an image of my brain, you're going to see where my abnormality is, you're going to focus the stimulation to my abnormal spot, and you're going to see if you can make it better. That would make sense, right? That's actually not what we do. So then you say, well, okay, well, you did all this science and you show me these beautiful maps with all the colors, so you know which parts are abnormal in depression, so you probably choose the right one from an average of some other depressed people's brains. And even though I don't have to go in the scanner, you know where you're aiming for me is related to this problem, and so you're going to focus on that place based on averages of lots of other people's brains. No, that's actually not what we do. And then finally say, well, at least you're looking at my head is much bigger than a very small person's head, and so if you want to hit a zone of the brain, then you're going to at least move forward a little bit more on my head because it's so much bigger than somebody else's. No, actually we don't do that either. So that's a problem. And I'm going to talk to you more about individualizing stimulation and where the hopes are for that, but just to give you a very brief overview of transcranial magnetic stimulation. So you plug it into a regular kind of wall socket, there's a magnet and something the size of a desktop printer. It fires electricity through this little coil that's resting against your scalp. It causes fluctuations in the magnetic field that carry electrical energy into your brain and makes a little stimulation at the place where right in the middle of the coil. So it's pretty circumscribed to that area. You don't need anesthesia, you don't need muscle relaxants. You kind of walk into an outpatient clinic, you sit down, they rest it against your scalp, and they start doing the stimulation. It's as easy as that. It does feel like something because it hits your scalp muscles on the way down to your brain, but having your brain stimulated actually doesn't feel like anything. There's no seizure, there's really, you can kind of sit there, watch a movie, you can think, you're aware, you're fully aware of what's going on. So let's get back to this idea of individualized brains. So brains look different. Maybe to the untrained eye across the bottom, you say, oh, those brains all look about the same to me, but for neuroscientists, you look kind of closer and you say, oh, those are actually kind of different shapes, they're kind of different lengths, and they are actually quite different. And so if you go in for the approved treatment for treatment-resistant depression with TMS, the way they find the place in the brain to stimulate is they find a place where they do a little zap to your motor cortex, they get a hand twitch, and they move 5 centimeters ahead of that. 5 centimeters for me, the smallest person on earth. Like they exactly the same, right? So 5 centimeters for me is probably going to be in different real estate than for somebody with a really small brain. And that actually turns out to be true. They did a study kind of after the case and said, okay, you follow that 5 centimeter rule, and where you hope to be is in a place called the dorsolateral prefrontal cortex that we think contributes to improving the outcomes in depression. And actually there's a pretty sizable minority of people that clearly you are not in the DLPFC when you stimulate. But there's no individualization, you use the 5 centimeter rule, and that's where you get stimulated. And so really the brains are pretty different, probably just based on very gross anatomy, which is a very kind of low level from a neuroscience perspective. Your brain should get a different stimulation target depending on your shape and size. So this is a study, I ask you to focus on the top row. We're often chasing deeper brain areas when we're trying to treat anxiety and depression. One of the most prominent ones that we keep seeing in depression as being abnormal is an area called the subgenual cingulate cortex, which is the top left. The side view of the brain, that little red blob. So that one's extremely active in depression. What we'd like to do is try to ramp it down. When people get treated for depression, they often see that there's a correlation with how well they do from any treatment and that brain area going down, so we really want to focus on it. So what the neuroscientists do, they say, all right, we'll collect an average of a whole bunch of people's brains, we'll find the place on the outside of the brain that's correlated with that place, and we're going to aim for that spot. Because we hope, since they're correlated, that there's actually a relationship between them, and if we push on the outside, hopefully we're going to reach that deeper brain target. So if you do that, if you look at the leftmost column of the sideways brains, that black circle is the average spot where you get the most connectivity, and you want the patch of brain underneath it to be blue, because that shows the relationship that you want to the deeper brain area. So on average, that black circle falls in blue territory, you're good to go. But if you look at subject number 1, which is the middle column, again, that black circle falls in blue areas, so you're good to go for that person. But then you take another person and you bring them in, you scan their brain and you look, and there's a bunch of red in there. And having red in that black circle means that the relationship is opposite, which means presumably if you add energy through that circuit, you're going to push that brain area up instead of down. So it's definitely problematic that people's brains are not the same, their functional patterns are not the same, and we think that has to do with how well the treatment works. So if you look at what's really wonderful about psychiatry and all the brain imaging experiments is now we used to have studies of 10 or 12 people. Now we have studies of hundreds of people. One of those is a study that Yvette showed you the outcome from, a recent study that largely shows the same pattern as this study published a few years ago that had more than 500 patients. Aggregating all their data from a whole bunch of different labs and showing where the connectivity patterns are stronger or weaker for depression, which is awesome. And you feel like now we have this really clear evidence that there are huge patches of cortex that are talking to each other in abnormal ways. But it's also difficult to take that evidence and say, which one of these places is more important and which one should you focus on and which kind of symptoms correlate with this pattern versus another pattern. And we think we can understand a little bit more at least about the relationship between the brain areas and which ones are more important and which ones are driving the output of the other ones by focusing on single brain areas and stimulating them with TMS. And what's really wonderful about new kind of technology and applying TMS is that I can put you in a scanner, I can collect an image of your brain, and then I can bring you back and put a camera in front of you that links your actual physical presence in front of the camera with your brain scan so that I can move my TMS coil around over the top of your brain, watch it in the camera, and I know exactly where on your brain I'm stimulating, which we think is really useful. So you develop a bunch of targets individualized for that person from your brain scan data. I know where the hot spots are. I know where the cold spots are. Now I can start investigating. Can I push on the cold spots? Can I push on the hot spots? Can I affect your mood? Can I affect your symptoms? Can I affect your brain readouts of putting somebody in the scanner like this, which is what we do in a lot of the projects, because this is really important information of measuring for each individual person, when I stimulate this place, what is the response in their brain? And how does that relate to their symptoms and to their outcomes? So you can do things like you take a map, you start out with it, you say, I think I know where I'm going with this map from that person or from an average from other people. And then you stimulate it and you record in the scanner. And you can do neuromodulation, so you can influence that circuit by repeating a bunch of trains of stimulation, and then you can ping that circuit again and see what did I just do to that part of the brain and to its response in the rest of the brain and its communication patterns. And we think that testing this in this kind of regimented way will give us a map that's individualized for each person. Where is your kind of brain atlas compared to the average one, and how important is that? And then I make a new map. A new map is individualized for you, but we can also average it all together, and we can understand if people like you with your symptoms or with your disorder look like the average, or how different does it look. When you do this, you kind of interleave this pattern of stimulate, record, and you make a new map for that person, and you see the entire brain. So you know what part of the subgenual cingulate or amygdala, et cetera, that part of your brain talks to. We can measure that. And we did that by focusing on a lot of the fMRI studies that other people have done before us and saying, we want to stimulate, we want to go in at one point with TMS, but we think we can actually influence networks. And we found that we can get those networks talking to each other, and we can get them to push each other by just focusing on single brain areas and stimulating. And so from that early work, we found out that we actually can stimulate a whole network just by hitting one particular spot that's accessible to non-invasive brain stimulation. So we were excited about that. But like I told you earlier, some of the really important deep brain areas like the amygdala, hippocampus, subgenual cingulate that we think are really important for the disorder are deeper in the brain. And so how can we reach those with TMS? TMS just affects the surface. But because there's all this wiring kind of pattern already there, we think that we can influence deeper brain areas by focusing, by using your brain image to decide where to stimulate. And so we did that in a healthy group of people. We ended up, these are all the places we stimulated, and trying to find evidence that we can stimulate some of the deeper places in the brain. And so An initial question is, can we reach those deeper brain areas? Also, can we actually reach them in depressed patients? Because that's also important. If we can reach them in patients, can we actually cause a change in those circuits in the patients? And does that at all relate to the symptoms? So it's kind of a tall order, but starting with the first one, we found that in all these little red kind of blobs, we found that we can influence the amygdala, which is one of the targets especially related to anxiety. The blue boxes show that we can influence the subgenual cingulate, which is the target for depression. So all we're doing is hitting that surface part of the brain that's correlated with the deep brain area and recording, ping, record, and seeing if we can move those brain areas. And it looks like we can. This is a replication. The little manta ray looking thing in green is showing the subgenual responding to single pulses of stimulation, so it's pushing it down, which is the direction we'd hope, and then compared to stimulating a different area that we didn't think was related, which has a much flatter response. So we've shown it and then shown it again that we can actually reach that subgenual cingulate that we think is really important in depression. This is more recent data suggesting that there might be actually a spot further down on the brain that when we stimulate there, we can cause the amygdala activity to go down. When I showed you the earlier data, the amygdala was going up, which may not be the preferred clinical direction. So seeing that we have some control over where we stimulate and the effect that we have on the deeper brain area is also potentially really important. So in response to the question, can we reach those deeper brain areas, we feel like we have pretty good evidence that this non-invasive stimulation can affect the deeper brain areas that we think are really important. This is a study of depression. Trying this out in depression, we have this design where the people go in the scanner and we give them the single pulses to the brain area that we think will reach that subgenual zone. And then we have them go outside of the scanner and do something that looks a little bit more like a treatment. So they come in for 3 days of double dose TMS, and then we put them in the scanner again and see if their brains changed and if their symptoms changed. And it turns out, so this is the kind of engagement down here. This is moving the brain off of its baseline. So we have a pretty strong effect moving the subgenual in the patients. So this is what we've shown 2 times in the healthy controls. So this is great that it moves it in the direction that we think is clinically useful. We stimulate them across in the scanner again, 3 days of a mini treatment, and then do this again. And you can see this kind of trajectory of the response of that subgenual changing over time. And that's correlated with symptom improvements. So the people are feeling better, and that response to stimulation is changing over time. So in response to the question, can we reach those deeper brain areas? We feel like we can. Can we do that in depressed patients? It looks like with preliminary evidence that we can affect those brain areas in patients, and we can change the brain areas in patients and that they're related to symptom improvements. So in conclusion, with that section, we're showing that neuroimaging alone can be augmented. So we're using imaging, right, to individualize where we stimulate. So the imaging data are very important. They help guide where we do the stimulation. And if we follow each person's actual brain data and use it as a targeting scheme, then it looks like we're effective at reaching the deep brain areas that we care about. And then we feel like the new maps are guiding individualized treatment, and we're able to change brain activity in patients as well as healthy people. And then when we're successful in moving the brain target, we're also successful in changing symptoms for the patients. So really, I didn't show you a comparison of the standard approach, the 5 centimeter rule, compared to what I do using each person's brain data to guide the stimulation, but that's coming up. That's something that we're working on. And also, depressed people share something in common, but they're also different, right? People are not diagnoses. So for some people, having difficulty sleeping is especially important or especially bad, or concentration problems are especially bad. So there may not be one brain stimulation target that works to fix all of those symptoms. So it might be kind of related to what your symptom combination is and what your brain activity tells us, and following that evidence in terms of what we can influence and what we can change and what we can't, I think will be really important. Unlike other things that are more complex, like a psychotherapy or even a medication, we have a lot of control over where we go in with the changes we make to the brain. And so I think following this approach, we can kind of gather a lot of new evidence for what brain targets are the most important and to individualize them. Another thing we're potentially doing down in the future is using people's brain readouts in the scanner to say how well are we doing. So not just saying I'm going to look at your map and fire there for a target, but also playing around with the stimulation parameters a little bit online while we get recordings from your brain activity and saying, okay, can we tweak these so that they work really especially effectively for you, and seeing if that demonstrates better outcomes. And then there are a lot of things that you can do just with the equipment in terms of how you deliver the pulses, how you make them kind of more tolerable to people, how you make them shorter, so that the treatment outcome potentially could be better. So there's pretty straightforward, like equipment side things that haven't been tested rigorously that are available to us with the existing technology that we haven't tried. And then Yvette mentioned focused ultrasound, which is a great way of stimulating deep in the brain without influencing the surface. So you have a lot of control over focusing ultrasound deep in the brain. They use that to remove cancer cells deep in the brain. They also use it non-invasively, of course, to see a developing fetus. So the technology is pretty straightforward, but its effect on brain activity is still very controversial, and it's definitely being applied to humans, but understanding how it works and how it might induce a lasting change in humans is still very up in the air. I've been very fortunate with getting funding from National Institutes of Health, which means I'm thanking you guys for those of you who pay taxes because the NIH funds a lot of the work, the big work, but we have wonderful collaborators as well. I also ask that if you're a neuroscience student at Penn and you're very interested in this work, there's usually openings. If you're a member of the community that just wants to contribute and you don't have an illness, you can likely have your brain stimulated. If you're safe to be in an MRI, if you're a patient looking for something new to try, and you have post-traumatic stress or depression, and you're looking for something new to try with this individualized targeting approach, I hope you'll look us up on the web and consider spending some of your time with us. That's about it. Thank you.
Thank you for your talk. My question is kind of about the difference in the brain. So you said that kind of as you image the person, you might be able to kind of localize where you need to stimulate. So how long does that brain image and that kind of localization last, given like neuroplasticity or changes in that person's disorder?
Yeah. So the question is, how long does that map really tell you what's going on in that person, and should you focus on it? And honestly, we've scanned people with that same kind of sequence sometimes a few times, and we've seen that it lasts for, I'd say, on average, a month, where it's very kind of stable. After that, I think you need to, in our experience with very limited kind of looks into that, we would say after about a month we rescan people. But there's another student actually here at Penn who's interested in saying how repeatable are those scans over what period of time, and what are some of the contributors to the variability that you see for an individual person across multiple days. So that's important.
Hiya. Given that there are many substances, including drugs, that we know something about their effect electrophysiologically or synaptically, how do you see these effects in bulk regions of a brain in which there must be many types of neurotransmitters, inhibitory, excitatory, etc.? And then as a follow-up, what about what kind of electrical changes can you imagine are being invoked at that distance with all those dielectrics and short circuits and everything going on.
Yeah.
Doesn't that worry you?
Yeah, it's absolutely the case that that typical kind of chemistry that affects your brain's ability to change also affects your brain changes to TMS. It seems like everything you measure in that domain is kind of influences the output, and so from known mechanisms, but there's also kind of a complexity of layer-specific kind of brain contributions to the net kind of effect of whether your brain is active or less active in response to stimulation that I think we can't predict very well without actually testing it in the scanner. So some of the targets going up, some of them going down, right, that I showed you in one targeting strategy, the amygdala activity went up, and in another one it went down. It's very kind of hand-wavy to suggest that I know the mechanisms, but that's why I think it's really important to do the mapping studies where we say, okay, we tried to engage the circuit and hitting this spot, that spot, hitting a more active, less active spot, and seeing what the downstream effects are.
Well, do you think that there's ultimately a change in membrane potential somewhere?
Yeah,
How
I
big
think.
could that be?
Yeah,
Based on what you know of the physics?
it's stable enough that you can measure it not just acutely while you're online in the scanner, but you can measure it before and after a treatment. I think the changes I make in my data are a little bit more kind of short-lived, but when you start doing a real clinical dose of TMS, you get 4 to 6 weeks of daily TMS. I think the patterns tend to be more stable.
Okay, thank you so much, Dr. Oates. And thanks again to all our speakers. They will be available outside with the reception. And thank you for everyone for coming. Thank you.